Let's start with a basic introduction to birds, their classification, and evolutionary origins. So why study birds? One of the things is they're fairly easy to study. You can basically find them everywhere. You can find them in every single biogeographic region, so the diversity there is quite amazing, and within any area you'll get different habitats. You're going to have different communities of birds. And the other thing is, most of us, certainly talking about myself, I'm pretty diurnal. And so birds are diurnal, and so that makes it convenient to study them as an ecologist. And also, it's just undeniable that birds are pretty, and that's why birding is so popular. It is really one of the fastest growing outdoor recreation hobbies. They're very colorful as far as their plumage goes, and this leads to a lot of people wanting to take on the challenge of identify them to species. And what we're going to find out in the lab is one of the really best ways to identify them, though, is through their vocalizations, which can be quite diverse. So birds have had a very important relationship with humans for a long time period. They're very well represented in religious documents and as government symbols. So think about the dove in the story of Noah's Ark. There's also a raven in that story as well. But ravens are very important in poetry, like Edgar Allan Poe. And also ravens are very important, particularly in the Pacific Northwest Native American cultures. Eagles are very powerful symbols in Native Americans and Romans as far as power and warlike entities. Birds are very well represented in art, going back as far as some of the earliest cave paintings. And if you look at some of the mosaics, the Roman Pompeii, they're constantly finding mosaics showing bird depictions there, and also in Egypt as shown here. And some of these are in such good detail that you can actually identify them to species. And in music, there's some musical compositions in classical music that really are focused around birds. So, for example, the Magic Flute of Mozart, Aaron Copland's Appalachian Spring, if you listen to the opening movements of that, it's actually was derived from the idea of a dawn chorus, and we'll talk about the function of dawn choruses in birds. And also, birds are pretty delicious, and so we've domesticated them for food sources. Chickens are domesticated from wild red jungle fowl, which are originally from Southeast Asia. And this first domestication appears to have happened about 5,000 years ago. And about the same time, carrier pigeons were being domesticated by rock doves in Mesopotamia and Egypt. Mallards also have a long history of domestication, not quite as long, so about 3,000 years, and they're derived from wild mallards. Turkeys, we don't know the exact timing of their domestication, but we do know that it was occurring in Native American tribes before European settlement. From a scientific point of view, birds are interesting to study because in many regards they're very diverse. They're the most diverse terrestrial vertebrate lineage, and on second only in vertebrate diversity compared to fishes. There are about 10,000 living species, and there are a few new ones discovered every now and then. The exact number of species could be debated based upon your definition of species definition, but it's in that ballpark. And the first true birds were derived probably about 116 million years old, but we're going to talk a little bit about their relationships to other theropods and how actually defining what a bird is and where birds started, you could actually debate. Birds are very diverse ecologically. You can find them again in a diverse range of habitats, and in these different habitats there can be many species in that community due to their extremely variable foraging patterns. So we're going to talk about that with regard to both their digestive tract, but primarily associated with their bill shape and some of their locomotion within that habitat. Movement patterns, they vary from some that swim, those that are cursorial and non-flighted secondarily, but most birds fly, and we'll talk about the importance of flight in the evolution of much of bird anatomy and behavior and their physiology. Another way that birds can vary as far as movement patterns is seasonal movement. We'll talk about migration and the importance of that in many species. My main interest in birds is associated with their behavior. Birds show a great degree of variation in behavior as far as their social aggregation. So many of you know about territorial birds, like think about cardinals defending all-purpose territories. But many birds are colonial, living in dense aggregations, and some of the more interesting ones can be seen in the tropics, like these oripendula, these long pendulous nests. They're about 5 to 6 feet long. This also leads to different breeding systems. So monogamy is, at least social monogamy, is the most widespread mating system in birds, and that's closely tied to their parental care behavior. But we'll see that there is quite a bit of variation in mating systems, and also different patterns associated with their nesting, where they nest, how long they nest, number of eggs, size of eggs. And so we'll be talking about that. But as I mentioned, a lot of the avian anatomical and physiological adaptations that we'll talk about are linked to the demands of flight. Flight is going to require one huge selection pressure, and that is to be as lightweight as possible, so that flight is more efficient. But at the same time, you have to maintain a large amount of powerful muscles to make flight happen, and also have a very strong skeletal system. So there's going to be some trade-offs here. So we'll talk about some of the placement of the bones in the pectoral girdle, like the very strong coracoids, which provide these arch-like or column-like support, these furcula, which are created by the fused clavicles, this massive keeled sternum, which serves as the attachment point for the most important flight muscles. So they have some pretty substantial bones, but I almost use the term solid but that's wrong, because they're not solid in most cases. If you look at the long bones of birds, so their leg bones and bones of the wing, you see that they're hollow inside, and they have these internal spongy-like structures that still maintain the support, but this greatly can reduce the weight of these bones. Some of the most foundational studies in ecology, evolution, and anatomy and physiology are associated with birds. Birds, because they're so visible, because they're diurnal, oftentimes can be used as an early indication of the health of a habitat or an ecosystem. And so birds are oftentimes studied. If you study the endangered species, for example, something like a red-cockaded woodpecker here on the bottom left, this is a bird that if it's present and it is maintaining a good population, you know you have a healthy upland pine savanna habitat. And we've been able to figure out what is required to keep that habitat acceptable for red-cockaded woodpecker, and if you make it acceptable for them, it's a pretty good habitat for most of the other community members associated with that habitat. We've also learned a lot about the evolutionary process by studying birds. We have some really important fossils. This is Archaeopteryx lithographica, which these fossils were just turning up after Darwin published his book on the origin of species, in which the topic of evolution first came to the front in science, and this provided some of the best evidence of what we call transitional fossils, lineages that show a transition from one basal lineage to a derived group of descendants. And then again, we also have learned a lot about things like the metabolism, feather architecture, respiratory structures, and the functioning of these anatomical and physiological systems by studies of birds, as we'll see. And the study of birds has not been restricted to scientists. There's a long history of citizen science projects associated with the study of birds. This can go all the way back to the Christmas bird counts. I just finished doing a Christmas bird count with Dr. Ron Thill and Cliff Shackelford. We do this every year. And these have been going on for well over 100 years in the U.S., and it's allowed for this long-term accumulation of winter trends in bird populations. A similar type of citizen science project is associated with the breeding bird surveys. So it's basically a way to keep track of long-term trends in breeding bird populations. And the more recent and really highly effective way of gathering data is through eBirds. This is an app that I'm going to encourage you to get on your phones as we go out in the labs and when you go out into the field on your own. It allows you to report all of your bird sightings, and it records time and location, and this is all uploaded to a website. And it allows for the Cornell Lab of Ornithology to track the movement of birds. So, for example, here's looking in 1 year the arrival and movement of orchard orioles in the spring, their establishment in different populations for breeding, and then their movement back during the fall migration back down into the tropics. As mentioned previously, there are about 10,000 species of birds, and we can understand how they're related to each other and classify them using two different approaches. Kind of the old approach is the Linnaean hierarchical system. We usually talk about kingdoms and class and order and family and genus, species, so forth. And you can still find those types of classifications for birds, but there are some problems with that. And part of that is due to the fact that these do not always follow evolutionarily comparable levels. So, for example, the class Aves and the class Reptilia don't make any sense as being comparable names as far as hierarchical levels, because birds in the class Aves are actually within the Reptilia, as we'll see when we look at it from a phylogenetic perspective. So we can still use some of these names, but by putting them in a phylogenetic context, it actually is going to make more sense and be more meaningful. So here we see the part of the phylogeny of birds, and you can see we've kept some of these same names, like the Charadriformes. These are many of these you would refer to as shorebirds. So that oftentimes is referred to as an order, and it does represent a nice monophyletic clade. So all of the descendants come from a common ancestor. And we can talk about clades being nested within clades, but we're always shooting for identifying monophyletic groups or clades. I want to talk a little bit about how you make phylogenies, but before we do that, I want to talk about where we get the data. So, and the importance of scientific collections. So at the university, we have a museum collection of vertebrates, including quite a few birds that represent both museum skeletons, but primarily skins, as shown here. Most modern museums these days also keep tissue repositories where they can preserve DNA, because that really is the most common form of data that is used these days in reconstructing phylogenies. So clearly the importance of scientific collections is associated with studying this DNA and also doing what are called morphometric studies, looking at changes in shape and anatomy and physiology of organisms, primarily anatomy. These can include things like looking at long-term studies of changes in, say, distribution, so we can see where all of these birds were collected and how that distribution may be changing through time. Importantly, also morphology and how morphology may be changing through time. Also genetics, looking at the genetic diversity of organisms through time, and are rapidly shrinking populations losing genetic diversity compared to older populations that existed at a time period when they were larger and had more genetic diversity. We can test that by keeping museum specimens that sometimes are hundreds of years old, and we can actually get some DNA out of the skins themselves, but it is easier out of some of the fresh tissues that have been preserved in the DNA repositories. Some of the morphological studies that have been really interesting include this species right here. These are house sparrows. House sparrows are introduced into the U.S., and some of the classic studies indicated how different shaped birds can survive challenging winter blizzard conditions by looking at the anatomical differences between those that existed in a population before a blizzard and those that survived the blizzard versus those that died during the blizzard. My former major advisor at the University of Kansas for my master's degree studied house sparrows as well, and what he did with Robert Solander from University of Texas, they studied how house sparrows in different populations throughout North America have adapted locally to their climates. And so up north in Canada, the sparrows are much bigger, chunkier, relatively short appendages, and this is adaptive for colder climates. But as they spread from their origin in the northeast of the U.S., when they are introduced from Great Britain, they have spread throughout the rest of the country, including the South. And here they're actually quite small, relatively thin, and relatively long appendages. And this makes sense for those types of climate. There are also changes in color. In open, sunny habitats, they tend to be lighter in coloration. In more wet, mesic situations, they tend to be darker in coloration. And there are adaptive reasons for this as well. But see, we wouldn't know that, we wouldn't be able to track those changes if we didn't have museum collections so that you can go and study those. An underappreciated benefit of having museum collections is they serve as a reference collection for artists. So I've had several artists, including those making sculptures and drawings and paintings, come and use the collection. And they've produced some really remarkable work, including a recent graduate student that did some work with the collection and really has come up with some fantastic stuff. All right, well, once you get the data, what are we actually looking for? What kind of characters can inform us about the evolution of a group of organisms? Well, when you're reconstructing phylogenies, what you're doing is you're looking for patterns of specific characters that will show evidence of a group of organisms being greater or lesser related to each other to degrees, right? Our goal is to find a group which is called a monophyletic group. And a monophyletic group is defined as one that has a common ancestor and all of its descendants. So what we got to do is we got to find a character that will indicate where those monophyletic groups exist. So we have to find some criteria of membership that says, all of the members of this group have this trait and they got it from their common ancestor. So here's a simplified phylogeny here, looking at two sister groups, the Crassiformes. So this includes things like kingfishers and bee eaters, and the Trogoniformes, which are the trogons and the resplendent quetzal in Central and South America. And if we want to look at characters that support these as being monophyletic groups, if you look at their feet, we see that Crassiform birds have syndactylous feet. All of them have this, okay? And the logical inference there is, why do they all have it? Well, they're all derived from a common ancestor that initially evolved a syndactylous foot. In the sister group, the closest related group, the Trogoniformes, they all have what is called a heterodactylous foot. And again, the reason they all have this, I guess we could assume that they just all independently evolved a heterodactylous foot, but that doesn't make a lot of sense. That's not what we would call a very parsimonious solution to explain why they all have this foot. A more parsimonious, logical conclusion is that they have a heterodactylous foot because they all inherited it from a common ancestor. So these types of characters that I'm talking about here, the syndactylous foot for the Crassiforms and the heterodactylous foot for the Trogoniforms, are what we call synapomorphies. This is what we're looking for. A synapomorphy is a character that shows us that all of the members of a group do represent a monophyletic group. So let's break this word apart to help you understand why it's called a synapomorphy. Syn means together, apo means change, morphology refers to the character itself. So we're talking about a changed character in an ancestor that holds a group together. That's where the syn comes from, right? So the fact that all Trogoniforms have a heterodactylous foot is because they all got that from their ancestor. Another term for a synapomorphy is a shared derived morphology. Okay, so in this case, the morphology is the heterodactylous foot. They all share that because it was derived. It originally was formed in their ancestor, but they all share that from that common ancestor. So that's the logic associated with the formation of phylogenies. And you can use morphological characters like this. And let's just kind of go through an example of how you use parsimony to guide you in making logical decisions to reconstruct a certain branching pattern. We call that a tree reconstruction. So let's say that we have these three birds and they have three characters of interest. Okay, so they have a crest or no crest. That's what the C represents. So if it has a C, that means they have a crest, like A and C here, species A and C. If it doesn't, like B, it has a 0. Okay, that means it doesn't have it. So we're talking about what we call binary characters. It's just one state or the other. The other is, look at the bill shape. It has a hook, represented by H in A and B, or C that doesn't have it. And the other is, do they have a webbed foot or not? All right, well, let's look at this distribution of character states among the characters of these three species. One possible interpretation is, they just all evolved these independently. Okay, so they all started with an ancestor that didn't have any of these traits, and they just all independently evolved a crest, a hooked bill, and webbed feet for those that have it. And so you would have just like this bush, really, instead of a phylogenetic tree. I guess that's possible. How many evolutionary steps would that take? It would take 1, 2, 3, 4, 5, 6. Okay, so each of these bubbles here represents the independent evolution of that character in that lineage. So it would take 6 evolutionary steps to get there. Well, what about this pattern right here? Maybe A is more closely related to C because the crest is a synapomorphy. All right, so the C here was a shared-derived character or a synapomorphy that evolved in the common ancestor of A and C, and that's why they both have that. Okay, well, if you assume that topology, it then requires you to have the independent evolution of the hooked bill and webbed feet in A and B. So if we compare the number of steps here, we had 6 initially. Now we have 5 steps. So yes, this is more parsimonious. This is a more logical reconstruction from assuming the phylogenetic bush that we started with. But is there an even more parsimonious solution? And sure. Let's consider now A and B as being sister species, more closely related to each other. There's stronger evidence of that, right? If we assume that the hooked bill and the webbed feet are synapomorphies, that's 2 synapomorphies supporting that. So that it's like it's got more voting power for A and B being a monophyletic group instead of A and C being a monophyletic group. It just had 1 vote. And the consequence of this is that now we only have to come up with a more complex explanation for the evolution of the crest. So this only takes 4 evolutionary steps. So that's the logic of parsimony. You're trying to come up with a way to explain the branching pattern that explains the simplest way that the characters could have evolved. So it's clear, like in some cases, some characters are ones you want to pay attention to and some aren't. Sometimes characters may look the same superficially, but they're really not, you're not comparing apples and apples. So that is due to what is oftentimes referred to as a pattern of convergent evolution. If you have the same selection pressure, sometimes you're going to evolve very similar solutions, morphologically and physiologically and behaviorally, to those adaptive challenges. So if you look at birds like loons and the Gaviformes, cormorants and the Suliformes, and mergansers and Seriformes, closely related, they're basically ducks, well, they look kind of similar. The way they sit in the water, some of their bill shape is similar-ish, but that's because of the same habitats that they live in and similar diets and adaptive morphologies for living in those environments and going after those food resources. But these are not really that closely related. They're in completely different clades, and they have independently evolved these morphologies that just sort of on the outside look similar. But if you look at many other characters, both DNA and morphologically, you can see, oh no, loons are different. They form their own group, pseudoforms form their own group, and so do anseriforms. So you have to look at all of the characters combined and look at the voting patterns. So the way they sit in the water is just kind of a one similarity that they show, but there are more characters that vote that they really are representing different clades. So you can use morphological characters, but these days there are far more powerful phylogenetic analyses that use molecular characters, either proteins and amino acid sequences, or in many cases actually looking at the nucleotide sequences in lots and lots of genes at the same time. And it has been a very powerful tool in helping us to uncover the degrees of relatedness of birds in different clades at different levels. We'll be talking about this more in the next lecture, and it'll be the focus of two of the labs that we're going to have early in this semester. So why do we even build these phylogenies? Well, one is so we can just kind of understand which groups are related to which groups and their basic pattern of evolution. But we can use phylogenies as a tool to understand other things like habitat associations or rates of evolution. So this is a study that looked at how quickly different clades were diversifying to try to understand what are the factors that make some groups more successful than others. And evolutionary success sometimes is measured by the rate of diversification. So how quickly you produce new species and avoid extinction. So just to contrast a couple of groups here, look at the paleognaths. The paleognaths represent primarily the ratites. So think about the ostriches and the emus. These are really old groups that some of them have survived, but they really haven't produced a lot of other species. So really low diversification, but long branches. Contrast that here down to like the New World warblers, New World blackbirds, and buntings that are really short branches, but lots of yellow and red indicating high rates of diversification. So they're constantly speciating, producing more and more species, but this has happened relatively recently. These branches don't go very far back. They're a relatively young group of birds. So see, a phylogeny is a way that we can compare these relative rates of diversification. Well, let's dive a little deeper. Instead of just looking at the phylogeny of birds, where did the birds themselves come from? And as I mentioned before, birds are reptiles. In fact, birds are dinosaurs. More specifically, they are the living representatives of dinosaurs. So if we look here, birds fit within the Theropoda clade. They are theropods. A subclade within that is the Maniraptora, and then what are usually referred to as the true birds are the Aviali. And many of those that are represented in this group are extinct lineages that we have pretty good fossil records from now. There have been some recent finds in China that really greatly increased our knowledge about the evolution of birds from their non-avian dinosaur ancestry. If we look within that Maniraptora lineage, one of the things that we've learned is where you actually cut off the Aviali and say, oh, these are true birds and these are Maniraptorian lineages that have bird-like features, it's kind of arbitrary, right? We're finding that feathers, for example, extend well into the theropods, maybe even predate the theropods. But well-defined feathers are seen in the Maniraptoria lineages in addition to those seen in what we would consider birds. And some of these could probably have weak flying or gliding flight as well. So this was a very gradual evolution of bird-like features into what we consider today true birds. If you look at the living reptiles, and this is one of the things that confuses people. People say, what are the most closely related group of reptiles or birds related to? Well, they are dinosaurs, right? So they're related to the extinct dinosaurs because they are dinosaurs. But if you exclude all of the extinct species and you only consider lineages that are alive today, then we have to jump way up here to the crocodilians. So the crocodile and the Nile crocodile and the alligator. This lineage here, because everything right here is extinct. And so if you get rid of these, the birds would collapse into this branch being most closely related to this branch. So if someone asks, what are the closest relatives of birds of all time? Well, it's the extinct dinosaur lineages. But of the living reptiles, non-avian reptiles, it would be the crocodilians. What are some of the traits that birds show with the other non-avian theropods? Well, feathers, as I mentioned, are found throughout this group and may even be found in the sister group to the theropods, so it may predate the theropods. Pneumatized bones, I mentioned earlier that the bones are lightweight, they're not solid, they have these hollow areas. This both reduces weight, but we'll see is also an extension of the respiratory system, and this predated and may have been a pre-adaptation for both flight and the evolution of the more efficient respiratory system we'll talk about in birds. Birds and dinosaurs had relatively small genomes with little DNA content. The upright and bipedal locomotion goes way down into the theropod clade. They also shared rapid development, calcified eggs. Social and nesting behaviors also goes way down into the theropods. And there's the potential that endothermy was found deep within the theropod clade as well. That last point is actually an area of quite a bit of study now. There was just a paper published in 2020 looking at the evolution of body size and how that may have related to the evolution of metabolism, respiratory systems in birds. So it's still kind of an area of study right now. But there is growing evidence that endothermy goes fairly deep into the theropod clade. I've already mentioned the importance of this specimen on here. This is Archaeopteryx lithographica. There are several specimens of Archaeopteryx that have been found. They're found in Germany, in an area of Bavaria, in these really fine limestone deposits that are about 150 million years old. And the fine granularity associated with these deposits has allowed for remarkable preservation of details. So we're going to look at some of the characteristics of Archaeopteryx, but I just wanted you to focus right now on the level of detail in the wings and the tail. There is such good preservation of these fossils that we can look at the detailed structure of the feathers, and they are identical in structure to modern day bird feathers. Skeletal characteristics. Archaeopteryx had a relatively small sternum, no keel, as seen here, in contrast to something like a pigeon that has a very large sternum with this keel that sticks out forward. That we'll see is important for understanding the potential flight capability of Archaeopteryx. Modern birds, the large keeled sternum allows for the attachment of the large muscles needed for powered flight. So this indicates that Archaeopteryx probably had weak flight at best. Archaeopteryx had teeth. Modern birds don't have teeth. Archaeopteryx had a long bony tail composed of multiple caudal vertebrae. Again, that is not something seen in modern birds. It has been reduced to this fused region of reduced caudal vertebra called a pygostyle. There was no uncinate processes, so these are interconnecting areas associated with the ribs, shown in number 6 here in the pigeon. You didn't see that level of connectivity associated with the ribs of Archaeopteryx, which is another indication of probably weak flight capabilities. Uncinate processes help to maintain some stability associated with the rib cage so that when these powerful flight muscles contract, it doesn't crush the thoracic cavity. A bizarre characteristic of the wing of Archaeopteryx, you may have noticed in the previous figure, was the fact that they have functional claws on their hands. Modern birds have most of the fingers fused into a structure we generally refer to as the manus. And Archaeopteryx had a relatively small brain case compared to modern day birds. And finally, there is no notarium. The notarium is a fused region of thoracic vertebra. We'll see in the lab. Archaeopteryx didn't have this as well. And again, that's just another way that there was greater support. A notarium provides greater support associated with powerful flight contraction, muscle contractions. So I've kind of highlighted the skeletal characteristics that kind of suggest that Archaeopteryx didn't have powerful flight, but there's one really important factor that does indicate that Archaeopteryx had weak flapping flight or at least gliding capabilities. If you look at the wing feathers of Archaeopteryx, they're asymmetrical, like you would see in this corn crake above. When you have an asymmetrical feather like this, this is indicative of a feather that was involved in flight. It is a more aerodynamically capable feather for creating lift. Non-flighted birds today, like the weka here, have symmetrical feathers like this. And so this is strong evidence that Archaeopteryx did have at least some flight capabilities. Some other important fossils that have indicated some of the transitions of non-avian theropods to birds is Confucius ornis. This is a more recent fossil. It's about 120 to 135 million years old. Still doesn't have a large keel, but it has a pygostyle now. So there is no long caudal vertebrate trailing behind that would reduce weight, make it more efficient for flight. And instead, they have these really long tail plumes, which indicate that when you see these in birds today, it's usually associated with sexual selection. And these were found in relatively large deposits, indicating that they lived colonially or in groups of some cases. And so it tells us a little bit about their sexual system. Confucius ornis also had separate claws on its wings. Okay, the origin of modern birds may have been as early as 90 to 130 million years ago. This is from looking at molecular clock evidence. So if we look at the rate of mutations in specific genes used to construct phylogenies, we can construct what's called a molecular clock and estimate divergence times associated with different lineages. And this evidence suggests that modern birds originated about 90 to 130 million years ago. This does not agree with fossil evidence. Fossil evidence is actually lacking. Modern birds' fossils do not go this far back, but fossilization is a tricky process. And so there definitely could have been birds back then, but the chance of fossilization occurring and then the chances of us finding those fossils is very difficult, so that's not too surprising. So this time frame indicates that modern birds evolved before the dinosaurs, the non-avian dinosaurs, went extinct. But it wasn't a very diverse group, so there were just a few birds were not very diverse until after dinosaurs went extinct. So some of the lineages that did go back this far would be the Paleognaths again, so the ratites I mentioned, ostriches, emus, also includes the tinamous, the moas, extinct elephant birds, but they did make it into the tertiary. But it also includes three groups of neognaths, so the anseriforms and the galliforms, and then a group we just generally refer to as the neoaves. So some of the early groups that would greatly diversify after the KT boundary. So again, it's likely that the extinction of the non-avian dinosaurs at the KT boundary about 65.5 million years ago provided the spark for the diversification of modern birds. So with the death of the non-avian dinosaurs, there were vacant niches to occupy, and this led to the diversification of both birds and mammals. And so after this time period, that's when you see the rapid appearance of most of the existing extant orders or major clades of birds today. And interestingly, we find that most of these were widespread globally, even when we see that they're regionally restricted today. So for example, hummingbirds are only found in the New World, so North and South America. Same thing with what we call now the New World vultures. But you can find fossils of these in Europe, so indicating that at one time they were more widespread. So this diversification was fairly rapid in most of these early lineages. There is a distinct lack of passerine fossils. You don't see them commonly until about 5 to 23 million years ago. So this is, we'll see in the lab, the order Passeriformes, or the clade Passeriformes, by far the largest monophyletic group of birds, but their diversification has occurred in more recent times compared to some of these other non-passerine orders that we'll talk about.